I would like to thank you, Daur, Sam, and Sarah for this opportunity to present my work at ISHR webinar series. So today I'll be talking about the role of transcription factor EB in the cardiomyocytes, especially in the context of obesity and diabetes. Okay. So as we all know that there are many mechanisms have been put forward to understand the obesity and diabetes related heart failure. And our laboratory focuses on one of the mechanisms which involves the abnormal cardiac metabolism to understand these complications. So to simplify the mechanism, during obesity and diabetes, there is a lack of glucose utilization causes hyperglycemia. Simultaneously, the fatty acid overutilization causes steatosis, and this condition culminates into glucolipotoxic stress in the heart. And furthermore, the glucolipotoxicity causes ER stress, mitochondrial dysfunction, as well as our laboratory has demonstrated that it impairs lysosomal autophagy and function in the heart, likely causing cardiomyocyte injury and heart failure. So before we understand proteotoxicity, we first need to know how proteostasis is taking place in cells. So proteolysis by lysosomal autophagy is one of the pathway which ensures protein quality control in cells, and it is stimulated in response to stress and starvation, as well as on the other hand, it is inhibited in the presence of nutrient abundance and in certain pathological conditions. So there are basically three types of autophagy: macro, chaperone-mediated, and microautophagy. And in any form of autophagy, since that eventually culminates into lysosome, so not only the proteins involved in autophagy, but the content of lysosome and the function of lysosome is crucial to ensure proper activation of autophagy. And in fact, these processes are tightly regulated through a master regulator called transcription factor EB, TFEB, which is a protein which was identified by Bellavio and others in 2009. And as the name suggests, it's a transcription factor localized in the nucleus, which is further activated in response to starvation and stress, and then translocated into the nucleus. At the other hand, it can be inhibited through its phosphorylation via mTOR and ERK. So once it is in the nucleus, it increases the transcription of genes involved in various biological processes, which involves lysosomal biogenesis function, as well as autophagy. It also regulates the pathways of cell survival, DNA damage, and most importantly, it has gained a lot of attention for its role in regulating cellular metabolism in various organs. So the question we asked was, does glucolipotoxicity negatively regulate TFEB to impair lysosomal autophagy and function to cause cardiomyocyte injury? And to answer this question, we first needed to know what happens to the level of TFEB in obese and diabetic heart. So we were fortunate enough to be a part of OPUS clinical trial, which is held at the St. John Regional Hospital here. And this clinical trials involve patients who had undergone cardiac surgery. And for our purpose, we actually categorized these patients as per their BMI into non-obese, pre-obese, and class 1 obese group. And then we received the atrial appendage from these samples, and we examined the TFEB protein expression. So we found that TFEB levels were significantly decreased compared to pre-obese and non-obese group. Most importantly, we also found that the level of TFEB were negatively correlated with patients' BMI. So, and not only that, but we were able to recapitulate our findings from clinical study using rodal models of diet-induced obesity, where we fed C57 black 6J mice with high-fat, high-sucrose diet for 16 weeks. And then we further examined the protein expression in the heart of these mice. So we found that the mice who were fed high-fat, high-sucrose diet, the heart of these mice had significantly lower nuclear TFEB content. On the other hand, they had a high levels of inhibitory phosphorylated form of TFEB, as well as the increase in the ratio of phospho to TFEB. And the low nuclear TFEB content further reflected in suppression in cathepsin B activity, which was used as a readout for lysosomal function. So together, we found so far that in obese and diabetic heart, there is an increase in inhibitory phospho-TFEB, resulting into decreased nuclear content of TFEB, which triggers, together, this triggers lysosomal dysfunction and cardiomyocyte injury. So next, we definitely wanted to know what if we can actually restore the TFEB levels in the nucleus and whether we are able to rescue this pathology. So for that, we developed adenoviruses which overexpress human TFEB and where it is mutated at serine 142 by alanine. And this will eventually result into the constitutive activation of TFEB in the nucleus. And we used neonatal rat cardiomyocyte and we overexpressed it with adenoviruses. And as you can see here that we were successfully able to induce nuclear translocation of TFEB when we infected the cells with adenoviruses. So then subsequently, we treated these cells with higher concentration of different substrates such as glucose, palmitate alone, and a combination of glucose and palmitate to simulate glucolipotoxic condition ex vivo. So then we first measured the Cas3 activity, which was used as a marker for cell death. And as you can see here that in CMV-GFP control group, Cas3 activity was significantly increased in response to palmitate and a combination of glucose palmitate treatment. Most importantly, the restoration of TFEB levels in the cardiomyocyte were able to suppress the palmitate and GP-induced Cas3 activity. And likewise, we further measured the cathepsin B activity, which was used to ascertain the lysosomal function. As you can see here that both in response to palmitate and GP, the cathepsin B activity is significantly suppressed. However, restoration of TFEB levels were able to rescue this phenotype. So to summarize, we found that TFEB play an important role in regulating lysosome biogenesis and function, as well as it play a central role in regulating cell viability. And having understood that TFEB is important in cardiomyocyte, we were curious to know that what other pathways have been regulated by TFEB when it is when the TFEB levels are low in obese and diabetic heart. And to find out more about that, we actually developed a cardiomyocyte specific constitutive TFEB knockout mouse model. And this is just a confirmation of TFEB deletion in the cardiomyocyte. And to find out that what other pathways have been impacted due to the loss of TFEB in the cardiomyocyte, we further performed transcriptome analysis. And interestingly, we found that out of 1,600 differentially expressed genes, majority of the genes were involved in the pathways related to cellular metabolism, apoptosis, cell death, lysosome autophagy, and cardiac function. And it was interesting to see that among the genes involved in cellular metabolism, 72% of the genes were related to pathways involved in lipid metabolism. So definitely we next want to assess the role of TFEB in regulating lipid metabolism in the heart. And most importantly, we observed that the transcriptional regulator of fatty acid oxidation, PPAR alpha, and fatty acid transporter, CD36, were significantly decreased in TFEB knockout cardiomyocyte compared to their wild type cardiomyocyte. And next, we wanted to assess the ability of the myocyte to form lipids in response to TFEB alterations such as overexpression or knockdown. So we overexpressed TFEB in the cardiomyocyte, and subsequently the cells were treated with either BSA or oleate, which was used to induce the lipid droplet formation. So as you can see here that in response to TFEB overexpression, the lipid droplet formation was significantly decreased compared to M-cherry viral group. And it was very interesting to see that when you actually knock down TFEB using shRNA, not only we were able to see increase in lipid droplet formation, but the lipid droplet size were increased further compared to its control shRNA group, suggesting that TFEB play an important role in regulating cardiac lipid metabolism. And since we know that TFEB is important in regulating metabolism, lysosome biogenesis, and cellular viability, lastly, we wanted to know what is the functional outcome of loss of TFEB in the cardiomyocyte, and therefore we lastly measured the cardiomyocyte contractility by using sarcomere shortening analysis system. And we found that we isolated the cardiomyocyte and we paced this cardiomyocyte at different frequencies ranging from 0.5 to 2 hertz. And we found that we didn't observe any change in sarcomere length, both at contraction and relaxation. However, the rate of contraction and the rate of velocity of contraction and relaxation were significantly decreased. And since calcium play an important role in cardiomyocyte contractility, we further measured calcium transient. And it was interesting to see that this TFEB knockout cardiomyocyte demonstrated decrease in diastolic calcium. However, no change in calcium were observed during cardiomyocyte contraction, and calcium transient during contraction were increased, and during relaxation were significantly decreased, suggesting that TFEB play an important role in regulating cardiomyocyte contractility and calcium transient. So together, to summarize, we found that TFEB is important in regulating lysosome biogenesis and function, cell viability, as well as lipid metabolic pathways in the cardiomyocyte, and most importantly, it also regulates protein involved in cardiomyocyte contractility and calcium signaling. So with that, I would like to acknowledge my lab members, collaborators, my supervisory committee members, and my funding resources, and definitely ISHR for giving me an opportunity to present my work today at this series. Thank you.
This time I would like to congratulate all of our speakers, Purvi, Ali, and Casey, for sharing your extraordinary work. And at this time, we're gonna ask question. Anyone can ask question for any of our three speakers, but please add the name of the speaker at the beginning of your question so that we can correctly navigate the question to the speaker. So with that, the first question, it's for you, Purvi. This is a kind of technical question. This attendee is asking, normally when he tries the antibody for TF.
Looks like we might have lost Sam for a minute. So I'm just going to go ahead with the question.
NRVM, neonatal red ventricular myocyte.
Yeah, so that's a good question. Actually, there are various bands of TFEB. We did observe different bands such as at 50, 70, and 150. And because TFEB is also phosphorylate, so there is a migration pattern you would observe. So it is always good to use a gain of function or knockout approach to find exactly what band you are looking at and confirm that that band is important for your particular model. So by using that, we know that the band which appears at 70, that is the model, that is the band is important for in our model. So definitely it depends on the experiment and your approach, how you are looking into to confirm the TFEB band. And he also asked about the oxidative status in the neonatal ventricular myocytes. So I haven't directly looked into the oxidative status of the neonatal red cardiomyocyte. However, in the other model of DOX-induced cardiotoxicity, which induces a reactive oxygen species, and we did see that when you restore TFEB levels back, it does reduces the reactive oxygen species in the mitochondria. So definitely TFEB has an impact on oxidative stress also.
The next question is from Tamoya, and he's also asking Purvi if TFEB directly binds to the PPAR alpha promoter region to regulate its expression level.
So far, there is no evidence of TFEB directly binding to PPAR alpha, and as well as we haven't seen in our model that we haven't actually examined that if TFEB directly binds to PPAR alpha. However, one of the study did show that when you actually use PPAR alpha agonist gemfibrozil, which causes stimulation of TFEB, activation of TFEB in brain cells. But in terms of TFEB directly binding to PPAR alpha, no, we haven't checked.
Okay, so the next question is from Sina, and she's saying that given that obesity is often accompanied with diabetes, hypercholesterolemia, and coronary artery disease, has there been any work on the effect of these disease, respective drugs or TFEB?
So, in response to your question, so when we so some of the studies from the literature, there is when overexpression of TFEB in the liver as well as in the adipocytes, it has shown that increase in TFEB levels have improved the whole body metabolism and prevented the diet-induced obesity and insulin resistance. And definitely, so TFEB would be one of the therapeutic, it would be interesting to see, considering TFEB as a therapeutic target to modulate lipid metabolism in obesity and diabetes related heart diseases.
The next question is about measuring cardiac function. So ask if you measured cardiac function in your TFEB knockout mice, including ejection fraction, fractional shortening, EA ratios.
Yeah, that was actually one of the future studies, and then we are still working on it. Unfortunately, because of lockdown, this study is going to be delayed. But definitely, I'm very curious to see the cardiac function of TFEB knockout mice.
So the next question is also for you, Purvi. I know you just answered the functional part, but do these cardio-specific TFEB mice has any in vivo cardiac morphological difference, or do they have any kind of lifespan changes?
So we haven't seen any morphological changes at the baseline, but when we if we subject these mice for any kind of metabolic challenges or stress, we might see changes in morphology. We might see changes in the cardiomyocyte morphology. However, at this point, no, we haven't checked. I mean, like at baseline, we don't really see any changes in function as well.
Thanks, Purvi. The next question is also for you as well. Laura's asking, is there anything known about TFEB levels in patients suffering from ARVC as a condition strongly associated with cardiac lipid accumulation? And did you use left ventricular cardiomyocytes for your experiments? And did you ever compare levels between the different chambers?
So I am not aware of TFEB levels in patients suffering from ARVC, but in one of the study where they have shown that human cardiac macrophages in ischemia cardiomyopathy, they have observed changes in autophagy level, which was impacted by loss of TFEB, and when they overexpress TFEB in these mice after ischemia reperfusion injury, it did prevented ischemia reperfusion injury by modulating lipid metabolism. So that is one of the study I can correlate that, but I'm not directly aware of about the level of TFEB in ARVC directly. And yes, in your questions to compare TFEB levels between the right and left ventricular myocytes, no, we didn't actually compare between two.
So this is the question from Alicia to you, Purvi. Can you comment on the mechanisms of calcium dynamic alteration?
Yes, so from our transcriptome data, we did see some of the genes which were involved in calcium transient, such as L-type calcium channel, SERCA and NCX, as well as CAMK2 which activation of which has been implicated in increasing the intracellular calcium level. So, some of these important changes we have seen, like these genes are differentially regulated and that play a very important role in calcium dynamic. So however, like this is one of our future study to figure out what exactly is happening. We were also planning to do some ATPase, circa ATPase activity. So definitely that is one of the plan to figure out the mechanism of calcium dynamic in this mice.
Great, thanks. So Purvi, the next question's for you as well. Sarah's asking, what are you planning for your future TFEB related work?
Yeah, Sarah, I would love if you can help me to finish my future work. We definitely want to these were the studies so far at baseline level, meaning that this mice has not been challenged with metabolic stress or any other stressor, and therefore at baseline we are seeing that there is an impact in cardiomyocyte contractility. This mice are able to contract as comparably to wild type cardiomyocyte. However, they are less efficient. But if we are able to challenge these mice with support high fat diet to induce metabolic stress, we might see some changes, and that is one of the future study we are planning to do. And as well as some of the functional data we are looking forward to have for these mice.
So the next question is from Daniel Johnson, and this question is for Casey. Given the fact that you didn't see any acute effect of propionate in adult myocyte, where the dyadic structure is nicely intact, but did you see any, see it chronically in neonatal myocyte where the structure is not quite there? So the follow-up question, do you think there are also some contribution of propionate localization between the two fractions? And also the second part of his question is also, did you try acute treatment of myocyte which had been detubulated?
So I'll start with answering the last part. We haven't tried any acute experiments where myocytes were detubulated. I get what Daniel was saying about comparing the adult and neonatal myocytes. So neonatal myocytes don't have T tubules; they're not fully developed. I'm not quite sure whether there is a localization of propionate, only because it's a relatively big metabolite. So I think the changes we're seeing are related to changes in expression of proteins involved in the processes, like a change in expression of phospholamban or NCX or other proteins. So I don't think I've really fully answered your question, but I'm not quite sure. But I think the main thing is, we're probably looking at changes in expression rather than like a localized effect of propionate.
Great. So the next question is for you, Casey, as well, from Michael Shattuck, who's asking if you think the changes in the action potential are direct effects on ion channel expression, or are they secondary to changes in the calcium transient after changes in phospholamban?
I think Mike's trying to prepare me for my PhD Vivo. Thanks, Mike. I would say, given that the whole thing is about propionate being a very potent modulator of gene expression, I'd say it's more gene expression changes. And I think especially because of the way the action potential changed. So in the neonatal myocytes, for example, the action potential shortened. And what I didn't show was ECG data from the mouse model. And in fact, QT intervals seem to be shorter. So given those changes, I'd say if, because the transients are being prolonged, it's more likely, if it was affecting the action potential, then the action potential would be longer because there's more outside current. So I would say it's more a change in expression of the ion channels. And one experiment to do, which was planned, but before the closures happened, was to patch clamp the myocytes and record action potentials from the mouse model. Because unfortunately, adult myocytes, they don't like the flow volt dye. They die when you excite them. So.
This is the question for you, Casey, and it's from Caitlin. So she's asking whether PCC a null model, do you think there is a role for increased substrate in contributing to the enhanced histone acetylation beyond HDAC inhibition by propionate? The second question she has is there any evidence for histone propionation?
Yes, you've, I don't know what the saying is, you've hit the nail on the head or something. That is a mechanism which we're interested in. It's actually what we, yeah, you're right. It's the work we plan to do in the future. I don't want to say more than that.
That probably means now.
Oh, God.
All right. So the next, we have a comment, Purvi, that she can read on her own, but Lauren Brothwood is asking Casey, what about NCX function in the PA mouse myocytes?
That's a good question. So one thing I didn't show was I didn't show the full protocol for the calcium transients. So what we normally do is we electrically pace a cell and then we stop the pacing for a few seconds. And then we'll expose it to a bolus of caffeine. And what that does is it allows you to then determine the contribution of NCX to the transient. What we found was that NCX function was also reduced in these animals, in the myocytes. I just didn't show it for clarity.
So the next comment's actually very related to what you are just saying, Casey. Michael again wondering whether calcium transient will change the shape of AP in the mouse because of sodium calcium exchanger.
Okay, and Daniel's commenting as well via calcium. I'm sorry, man, I don't quite understand what you're saying. There's some other questions, so if you're happy to just clarify, I'll come back to that. Sorry.
No worries. We can, yeah, Phil, go ahead. If you guys are still on, feel free to like clarify any questions in the chat. The other question we have is from Sarah for you, Casey, about have you looked at mTOR signaling in your model given its role in histone acetylation?
So we haven't, but I think there's a literature based on this looking at mTOR histone acetylation, but in kind of neurological models, because PA is a multi-systemic disorder. It doesn't just affect the heart. So there are research groups around the world who are interested in propionate-induced acetylation in the brain, in neurons. But we haven't looked at that, and we haven't looked at that signaling pathway, no.
Okay, so the next comments, thank you, Daniel, to make it easy for both Casey and Mike to understand each other. So I have a question actually for Ali. I just want to put it here. Ali, in your modeling, the whole modeling situation, can you incorporate any non-coding RNA or, for example, microRNA? For example, in your specific example of isospecific signaling, can you include or incorporate microRNA signal in between two genes? Is there any possibility, or is just the transcriptomics?
Yeah. Currently, our model is mostly based on the proteins activations and or gene expression, but some of our signaling components are some changes in the level in the mRNA expression. So basically, yeah, we can add the mRNA to our model, but it needs, because there is some difference between the timing and other dynamics between the signaling components and mRNA, we should consider this timing. And if we have the data, enough data on that specific network, we can add to the model. Yeah, it would be possible.
Sorry, Sam, do you mind if I just interject there as well and just ask Ali another question?
Sure, please go ahead.
Thank you. I mean, I was just talking to Jeff, actually, Sorceman today, And I mean, it's, you know, it's incredibly complicated to create the models that you create. And it seems like you're creating models for cardiac myocytes, for example, hypertrophy, cardiac fibroblasts. Do you ever envisage a time when it's going to be possible to make these models even more complex and incorporate multicellular models? You know, for example, cardiac myocytes and cardiac fibroblasts together, possibly even endothelial cells, because we know there's interaction between these cells.
Yeah.
So what's your opinion on that?
Currently, we are working on that. One of the interesting result that we see on the ERK activity, that was the significant difference between ERK activity in the fibroblasts and the cardiomyocytes. And we are working on that to incorporate the cardiac fibroblasts in our model, because practically the overall goal would be preventing the heart failure. And to preventing the heart failure, we should consider all of them together. Yeah, because our lab was working on the cardiofibrillars network too. So there are many potential in combining the endothelial, the cardiofibrillars, and cardiomyocyte together. And we also can incorporate the growth mechanic and finite element models to model the 3D changes of the heart after for different stimulation and different contexts. Yeah. We are working on that. Is our future direction. Yeah.
Okay. So, but presumably then you have to come up with, well, you have to feed in new data sets, because presumably you can't just feed in the data from, let's say, cardiac fibroblasts, you know, culture in isolation, cardiac myocytes, culture in isolation. You probably need to basically do co-cultures and then presumably single cell, you know, RNA-seq or something like that from the co-cultures, and then to integrate these models. Is that right?
Yeah.
Okay.
Yeah.
Because you can't use the current data really that's based on single cell, can you?
The current data we have are specific to cardiomyocytes. So if we want to predict the exact response of the cardiac changes, we should incorporate, the cardiac fibroblast data into our model. But we need, for example, to have some new experiment maybe that we consider all of them together with the same experimental condition, because different labs have the different methods and different experimental condition that we should consider in incorporating the data together. Yeah. Thank you.
All right. We have another question for Casey. Sarah is asking if you've examined circulating levels of BCAA and BCKA.
Sure. So we haven't measured this ourselves. And we didn't develop this mouse model. It was established in 2013 by another group in the Mayo Clinic. They've measured things like plasma glycine, alanine and lysine, which is shown to have increased. But they haven't done the branching amino acids. I'd expect them to be higher in our mouse model because branching amino acids are metabolized as propanol-CoA. So there could be a higher level. But no, it's a metabolic screen is something we've done, and we're just kind of crunching through the data. That's a good question.
I think the next question or comments is from Michael, and I think he gave you a little bit of background. You can read that. But the question is at the end, kind of just very long-winded question about whether you have evidence for ion channel expression changes. Can you explain everything just by changes of phospholamban, PLF?
Sure, thank you for clarifying. So in the mouse model, we haven't done transcriptome screen yet. That was again something that was planned just before the closures happened. So I couldn't say for 100% certainty that the changes we see in the mouse are due to changes in ion channel expression, but yeah, I can't say for sure, but I think it's likely, just given the changes we saw in the other systems.
But sorry, Casey, can I just sort of again jump in here? Because you've done RNA-seq on neonatal cardiac myocytes.
That's right.
Right. So have you seen any changes in, let's say, potassium channel expression, for example, that might, indicate that there are changes in?
Yeah. So there were changes in. I guess I kind of went through a bit too quickly, but among the list of the differentially expressed genes, some of them were subunits for potassium channels, but they're not the subunits that you associate the big diseases with. And I think that's partly because they're neonatal myocytes. So that's why we're pushing to complete the experiment in the adult, and maybe we will see changes in the more well-known subunits of ion channels.
Thanks.
All right, Casey, the last question we have is also for you. Did you look at the protein translation changes in adult versus neonatal cardiomyocytes?
So I believe Dipsicler is asking about the proteome. We haven't screened the proteome in either the adult or neonatal myocyte. It's something we've planned as well. And yeah, as I just said, we've only looked at the transcriptome in neonatal myocytes, but the adult is planned as well. Thank you.
Great, that's wonderful. So we are really grateful for the speakers that have presented today, and we want to thank you for your wonderful science and for being here and sharing that with everyone. Feel free to reach out if you guys have additional questions for the speakers today. Feel free to reach out to them and ask them and continue the great discussion that we've had outside of this webinar.
Right. Well, I mean, first of all, I'll leave Sam as well. Thank everyone afterwards. But I'd like to thank you as well. I mean, I've really learned a lot from today's webinar. Some great presentations, and I also have to say we've got a first for the webinar series. We have our first cat appearing in the background. So Purvi, I think we've seen your cat on screen, which is excellent. That's what we want to see. It's a true lockdown webinar series. You can't have a lockdown webinar series without an animal in the background. So thank you.
He always cooperate. When I'm talking, he's always stay away and let me focus.
Excellent. Sam, I don't know if you want to say something as well, or?
No, I think Sarah kind of summed it up very well. So I just want to congratulate all of our three speakers, and I would love to see more of our ECI, MCI highlighted topics. But that doesn't mean that I'm voting for lockdown for a long time. But it's fun. Thank you, Deborah, and your team to organize this.
Yeah, thank you so much for the opportunity.
Thank you very much.
Thank you very much.
See you.
Bye, guys, and thank you to the audience, and hopefully see you all tomorrow again.